1, 2, 3.
So these two can cycle through before we so those two can cycle through before the other two, exactly. So I'm going to close these and share some people in this room for whoever has to speak. Make sure they get it. Anybody has any slides?
Maybe we'll give Alexander 1 more minute to show up and then get started. He's around because I saw him this morning. So. Good timing. So why don't we get started? So I'm Ramnik Xavier, and we work at the Broad and Mass General. We've got all the key people here. And before we get started, I thought what we could do is go around the room, everyone introduce themselves, and if any of you all are working on gut-related projects or funding gut-related stuff, maybe you can give us sort of a brief outline, because I think that's the first topic on the agenda. So,
is this working? Yeah. So my name is Alexander van Oudenaarde. I'm at the Hubrecht Institute. We work on development of new
can everyone hear you?
development of new technologies, and as the gut is one of our favorite systems.
I'm Kat Tajantanakis, and I'm at the Sloan Kettering Institute in New York. And I'm a developmental and stem cell biologist with an interest in the endoderm and the formation of the early gut tube.
Omer Yilmaz, assistant professor of biology at MIT and GI pathologist. And we're interested in diet and stem cells.
I'm Loyal Gaw from Johns Hopkins University, assistant professor of neuroscience. We work on enteric nervous system development and disease, specifically Hirschsprung.
I'm Katie. I'm a postdoc in the Regev lab, and I'm a scribe.
Joshua Levin. I'm at the Broad, interested generally single cell, but not necessarily gut, just here to help.
Gut is the most important organ.
Ram Dasgupta from the Genome Institute of Singapore. We're looking at heterogeneity and tumor evolution in the colorectal cancers.
I'm Matthew Stevens. I'm a statistician at the University of Chicago. I'm interested in clustering and understanding structure of complex data sets, and I'm here because gut is the most important organ.
Barbara Englehart. I'm at Princeton University. I'm a statistician and machine learning person looking at single cell data generally.
I'm Jessica Langer. I'm program officer in Crohn's disease at the Helmsley Charitable Trust. And we are currently funding an RFA, or announcing an RFA, have an RFA open for gut-related, Crohn's disease-related programs, and our deadline is November 13th. We're doing this jointly with CZI, and the portal's on their space, so search Gut Cell Atlas, put in applications. We're always looking for more.
Hi, Garabet Yeretzian. I'm the director of the Crohn's disease program at Helmsley, and you just heard the interest in the gut and Crohn's disease.
I was told this morning that they don't have enough applications, that 15, they'd like to get to 50, so hopefully there'll be a few people who will apply.
Andrew Farmer, I'm the head of research for Takara Bio USA. We work on kit development, a lot around sequencing, but also we work on stem cells and particularly differentiation of iPS lines into endodermal lineages.
I'm Natalie Robottom. I'm the editor for Stem Cell and Developmental Biology at Nature, and I'm interested in hearing what you can find out with this approach.
My name is Jonah Kuhl from the Chan Zuckerberg Initiative. Work on the Human Cell Atlas there. Before that, a cell and developmental biologist, and love the gut.
Hi, I'm Samantha Morris. I'm an assistant professor at Washington University in St. Louis, and my lab is working on single-cell lineage tracing and reprogramming to endodermal lineages.
I'm Aviv Regev. I'm here at the Broad and at MIT, and together with Sarah, we brought you here.
And I apologize, we will have to step out for 2 minutes and come back.
Yeah, well, Sarah Teichmann, welcome Sanger Institute, and Aviv's kindly introduced me.
Clay Fisher, I work with the DCP to help bring in data for most North American labs.
Yeah, Laura Clark, the DCP, obviously we're super interested in making sure we represent the gut data well in the platform.
Matthew Spear from UCSC. I also work on the DCP. I also work for the California Institute for Regenerative Medicine and the UCSC Genome Browser.
I'm Jody Hirshman. I work here at the Broad, working for the DCP on communications and outreach.
Hi, I'm Gabby. I work with the DCP as well, and I'm a user experience designer.
Hi, I'm Brendan Wesley. I'm in Ludovic Vallier's group, and I work on liver single cell.
I got one here. Omar Bayraktar. I'm at the Sanger Institute. I actually work on the brain, but I'm interested in large-scale spatial transcriptomic applications. And I'm interested in seeing if they could be applied to things like gut biopsies and disease models.
Hi, my name is Ko Hao Chen. I'm a research fellow from Genome Institute of Singapore.
Hi, I'm Kylie Daggertano. I'm here at the Broad as a product manager with the DCP. I'm always interested in learning from all of you, but I have a special interest in this breakout because my dad has Crohn's disease, so I like to bring back some science for him.
Hi, my name's Deanne Taylor. I'm at the Children's Hospital of Philadelphia and the University of Pennsylvania, and I represent the Pediatric Cell Atlas, where we're doing all the organs in kids, and also Hertzsprung's disease in the gut and IBD, very early onset IBD, and we're already in the process of generating single-cell human data for that.
I'm Shyam Prabhakar from the Genome Institute of Singapore, and I work on colon cancer single-cell analysis with Kok Hao and Ram over here.
Hi, I'm Sang Kim from Korea Institute of Bioscience and a computational biologist interested in data analysis.
Henk Stoenenberg, Radboud University, Netherlands, interested in single-cell epigenetics and gut.
Jerry Wood from the University of Leuven and the Sanger Institute, working on single-cell multiomics technologies and using those for somatic genetic variation.
I'm Oni Basu from the University of Chicago, and we just started a new project in creeping fat disease, so interested in hearing more about gut and gut diseases.
And I'm Alan Mullen from Mass General Hospital, and my lab is interested in early endoderm differentiation and fibrosis.
Okay.
Hi, I'm Aishwarya. I work with Anya Bourdeau and Aviv Regev on mostly kidney single-cell transcriptomic projects, but also getting interested in the gut.
Okay, so I'm sure I can remember all the names, but I think what we're supposed to discuss is what's happening in the field, what are some of the common challenges, how are we going to process samples that represent this huge organ from the, so the question is, how do you define gut? So most of the description here is small bowel and colon, but gut starts in the oral cavity and extends all the way down. And then, so maybe we could sort of open up the discussion on sort of standard practices, how one might come up with smart ways of profiling the gut. It's, since Barbara is here, about some of the spatial construction. Alexander can tell us something about epigenome or methylation within the gut, but an open forum. So why don't Alexander start and then.
Yeah, well, I also wanted to talk a little bit about the spatial aspect, but I think what's very interesting, I just was at the single-cell genomics meeting, which was a few days before this meeting, where there were a couple of very new insights in how you can reconstruct the spatial position of cells in the gut as well. Because of course when you do a standard single-cell sequencing experiment, you would just dissociate your cells, and the cells go through a sorter or go into the 10x machine, and you have no idea where these cells come from. So I think what of course the gut is, or the crypts are very special, is that they're very clearly organized. There's the stem cells at the bottom, and then they migrate upwards. So there's a very specific structure of these crypts that you completely lose after you dissociate. And I think one of the things I'm thinking about really actively is how can you somehow get some information on the neighbors. For example, if you have a cell, can you figure out what the neighbors were, or what can you reconstruct the spatial organization in the crypt? But also, of course, if you have crypts neighboring that crypt, there's a lot of interesting data that show that a particular crypt, if there's something wrong in that crypt, it will send out all kind of signals to the neighboring crypts that will behave very differently, even though they are completely wild type. So I think one of the things that I'm thinking about is how you can learn something about the spatial information, but still get all the depth of sequencing information that you get with single-cell sequencing. So there is a couple of things you can do. Of course, you can try to computationally infer it. It was a very interesting talk by Nicolas Rajewski, where he basically says, well, all these cells are dissociated from each other, but what we know is that there is some continuity in the organ, right? So you can piece it. It's a huge puzzle that just dissociated, and now you have to put the puzzle together, but you know that the pieces have to fit together. So there has to be continuity on the gene expression space for all those 6,000 genes that you measure. They have to kind of gradually go from one level to another. You know that wind is high at the bottom, so then the wind signal would slowly disappear as you go up the crypt. Can you piece together all these cells back onto their normal position? And that will be the easiest way, because then you don't have to do anything extra. You just basically take the data, and the computer will tell you where they came from. So this is kind of an ideal situation, but of course it's too good to be true, probably. So that's one way I was thinking about. Another way is to rather than dissociating the whole structure, you keep the structure partially intact. So you have a cell that is still connected to the neighbor, and now you sequence those little pieces, and perhaps you have a Paneth cell next to a stem cell, and you don't know that they're a Paneth cell and a stem cell, but they're two cells, and you sequence them, and you would see both markers. So you would say, hey, this was a Paneth cell and a stem cell next to each other. So you don't want to dissociate to single cells, but just in pairs, for example, and then you can reconstruct the whole structure. So that was one thing we can discuss if you're interested in this. Just how would you somehow retain some of the spatial structure, and specifically in the gut. This is really essential. Not only within the crypt, but also along, the coordinate of the gut. And that's something that we actually recently have been measuring on the epigenetic side. Is if you take single-cell epigenomes along, in this case, the small intestine, what I found in our experiments is that the most discriminative signal is not so much the cell type, but is where the cell came from along the intestine. So if you take at the one position a stem cell compared to the Paneth cell, they look very similar in terms of the methylome. If you go 1 centimeter downstream, the methylome changes a lot, and the Paneth cell doesn't resemble the Paneth cell that was 1 centimeter upstream. They're very different. So it seems to be, actually the epigenome seems to be very much dependent on where you are rather than what you are. And we don't understand this, but it's definitely in the data. So perhaps that's some food for thoughts.
So maybe staying on theme, maybe Barbara, some thoughts?
Yeah, playing off of that, I think the gut is really interesting to think about spatially. It's not alignable like heart or brain. or liver, and it's not completely disassociated, like you were saying, like immune cells. But there are very concrete patterns that are shared across individuals, and I feel like this is an organ or a system that's incredibly suited for Sarah Teichmann's sort of multi-scale analysis, right? Where you try and understand spatial resolution at the very broad scale from, like you said, from the oral cavity down to the end, and then within each of those pieces, you have to build in patterns and relationships across those pieces within each of those structures. Getting that right, I think, is very difficult, and I don't know how to do it. But hearing from people who actually go in and understand every piece of the gut would be very interesting, I think. Besides that, I think there are a lot of methods that need to be developed to connect the completely disassociated cells that we have with all the imaging data that we're getting, and understanding how it goes with this sort of structured spatial model of the gut that we have.
So we'll come to methods in a minute, but you had something to say? No.
I was just going to ask whether we could elaborate perhaps on imaging data and the different methods. So maybe that.
Yeah. So maybe we'll come back to methods, but others along the same. We had somebody from pediatrics who was interested in profiling the pediatric gut and some aspects of disease, but we'll come back to that. Any thoughts on some of the challenges?
You want me to talk about it? I'm the informatician, but I can tell you some of the challenges are for actually getting the tissue. For us, is getting the tissue in the right consent. And usually there's surgeries. Hirschsprung disease, there's surgeries, and then they get normal margins, and we want to know if normal margins are really normal. And that's a question we have for pediatrics, because it's hard for us to do biopsies just for normal tissue. So, what the community thinks of normal margins and like pediatric samples where you can get this kind of data, would it be normal or? I can't talk about any other challenges right now.
Well, others, so one of the challenges is how to sample this large organ. And the good part of the gut is that it's mostly reachable by clinical care that allows you to get to those sites. And so in many ways that's a plus. It's unlike the brain, a lot more accessible organ. And some of the principles that could be identified in the gut then could also be applied to other tissues which are not reachable.
I had another question regarding the disease context. Not only is the tissue where it comes from in the gut important, but the microbiome is also important. So I wonder if there is any thoughts of looking at the microbiome and the tissue at the same time.
Yeah, so I was still staying on normal, so let's try to get the normal aspects of the gut resolved, and then we'll get to disease. Obviously diet and the microbiome is going to significantly influence it, and that I'll hand over to Oma, who's thinking a lot about that. Normal gut, sampling the normal gut, protocols for analyzing gene expression, reproducibility, all of the above. We've solved all of these problems, or? Moshe, any thoughts?
Well, I think we need to do something very comprehensive. For example, as we discussed here, try to understand the proximal versus the distal side of the different zones of the intestine, including the small intestine and the large intestine from humans, if we can. That's a little bit of a problem in the small intestine, but I think we can overcome it with resections from patients, at least, that we have. It's adjusting tissue, or we can use postmortem samples if we can very fast. Other than that, I think we also need to do a very comprehensive and try to understand the, let's say, the crypt villi axis, try to understand what's going on there. There are already some papers in mouse, but we need to do it in human. This is it.
Yeah, so getting access to various parts of the gut is not a problem, because now there are new double balloon endoscopes that can get you all the way down from both directions. So I don't think And the other trick you can do is you can also deliver I'm a gastroenterologist, so I know this stuff. You can deliver a net, and that net can go further down to areas that the scope cannot get to, and you should be able to collect. The challenge, though, is you can only get mucosal sampling. So if someone is interested in creeping fat, then how do you get to things on the serosa, on the outside of the gut wall? And that's relevant to disease, but we'll come back to that in a minute.
Just a question out of ignorance. You have SOPs on timing for collection? I mean, whether somebody's been fasting and for how long, and how that affects the expression in tissue?
Yeah, so that, I think we can document that with regards to fasting. I mean, the challenge with the colon is how to sample the unprepped colon. And so that becomes a little bit of a challenge. Other thoughts?
It's a curiosity and thought, appreciating that the gut itself is quite complex, but then the interactions and communications with other systems, so nervous system, immune system, et cetera. And so to the extent that any profiling of the gut also has to be coordinated, or there's additional value of coordination with collections and profiling of matched samples from the same individual. How you handle that or deal with that and integrate the data that way.
In a particular that case, it's really important it's from the same location, right? Because they are talking to each other, right? So if you have mesenchymal cells and the epithelial, they have to be the ones from exactly the same location. So that's actually very challenging, but required. Or neurons that are firing at that location. You want to compare them to the epithelial cells from the same, 10 micron kind of microsphere.
I think the idea of keeping in mind the state in which the biopsies are taken is really important. So for example, many patients are fasted overnight, and we know that fasting can lead to dramatic, even a 12-hour fast can lead to dramatic gene expression changes. So I think it's important to keep that in mind and to obtain biopsies from other tissues under the same nutritional and metabolic states.
I'm wondering whether actually it would be useful. I assume often things are collected during fasting, but maybe it's also useful as part of an atlas to look at tissue not after fasting, but immediately after eating to know those differences, right? I mean, obviously you need to coordinate that in some way and standardize it, but.
Other thoughts? So I think broadly, I think the idea of how many guts to sample, what's the distribution of sampling, what sort of nutritional factors might contribute to sampling and readouts. I think that hopefully there's going to be consensus for all the other organs at the same, according to the same guidelines. Other thoughts?
For children, we're in the process of putting out a paper on this, but for the whole pediatric cell atlas. But one of the concerns that the researchers have is that the child's gut seems to increase in size over the first 5 years of life significantly. So how is that going to change the interaction, for example, between crypts or across the length of it? And apparently there's also different, obviously there's different, they have to maintain the function of the gut during those years while their colon and intestine are growing. And the other challenge we have is the microbiome seems to change a lot in the first 3 years of life significantly, and how that could be somehow managed and mapped. And this might be a problem for an atlas because you have temporal changes along the child's gut over ages, which is something, it's a longitudinal issue. And so
yeah, all good points. I think one of the challenges with sampling the normal gut, so one of the learnings of microbiome studies is that when we try to recruit patients as controls for microbiome analysis of gut-related disease, you select a phenotype. They often have sort of non-specific GI symptoms, and they want some information on it. So how do you find the true control? I think you had a question.
Yeah. Just going back to Moshe's point of postmortem sampling, what are some of the challenges with that, and why not go after postmortem samples since you have access to the whole system?
Yeah, so as part of the GTEx program, the plan is to go after postmortem samples. I mean, obviously, the gut is unique in many ways because it has a lot of protease activity. And so it's extremely sensitive to time of perfusion. The other is that generally most of the epithelium in the lower part of the gut is used to living in anoxic conditions. And so the minute you open it up, you sort of expose it to this wave of oxygen that potentially could change gene expression programs.
I mean, I agree with Ramnik highlighted all many of the important points, but the pathologist who's done many autopsies, the intestine breaks down immediately. Within an hour it starts to break down. So I think, if one were to focus on autopsies, they would have to be very rapid in nature to get useful data in the intestine.
Other thoughts? So I think, at least for part one, before we get to analytical methods, which I'm going to hand over to Alexander, I think all of us would have to come up with some consensus on how to collect samples, how to profile these samples, and the standard SOPs across different parts of the world. Obviously, very briefly, diet and the microbiome is going to be super important. And the question is, do we sort of sample the microbiome at the same time that gut samples are being collected? Even if you don't analyze them at the same time, at least you could go back to that sample set and then figure out what that might have contributed to. Other thoughts? Okay, it's yours. You're going to talk about analytical challenges in studying the gut.
Data analysis.
Yeah.
Yeah, I think the gut is probably as challenging as any other organ in terms of just analyzing single-cell data, but there are, I think, a few things I can think of that are specific to the gut, which we run into, for example, is that there is, the population of cells in the gut are quite variable in a sense that you have very tiny cells, like for example, at least in the mouse, that's where I have most of the experience, the LGR5 expressing cells. There are little slithers of cells that are squashed in between two huge Paneth cells. So if you would do single-cell sequencing, you have these enormous Paneth cells that have the volume that is 10 times the volume of their neighbor. So those cells are much easier to sequence. So you will see a lot of Paneth cells, but because you have 10 times more the material, right? So you have to be very careful there in not throwing away the cells that didn't work, quote unquote, because there's lots of very interesting cells that just are small and super important, because the stem cells are actually probably more important than the Paneth cells. So I think that might be, I'm not sure, there might be other organs that have the same challenge, but that's something that we run into quite a bit, is that if you're not careful, the proportions of cell types you find in a single-cell sequencing experiment might not be reflective of what you see if you just take a section, frozen section, and you count the cells. And this has, for example, the consequence of this is this huge Paneth cells that have a lot of RNA. They also have very highly expressed markers, so you can really easily pick it up. All these defensins and they scream at you, so you can almost not make a mistake with a Paneth cell. You just need a few transcripts and you know it. Whereas a transit amplifying cell, it has kind of a little bit of a vague transcriptome, and this a little bit higher, a little bit higher. So and they're much smaller. So that's something you want to be very careful with. And I also have the feeling that certain cells just don't like to be dissociated at all. And I think the goblet cells are particularly sensitive to this. So if you're not careful, most of the goblet cells will not survive the procedure, and then of course you won't see them in the data. Was there a question? Or no? You were just okay.
I wanted to ask you a question about the Paneth cells' big size.
Yeah.
Would single nuclei sequencing bypass?
Yeah. That would help. Of course, you would lose a lot of information from the cytoplasm. I think you can live with it, but you have to be careful. So you just have to realize that some cells have a lot of material and some cells don't. And often, you can say, well, you use spike-ins, for example, and that is actually typically not working somehow, because we found actually that the spike-ins behave very differently in a lysate of a Bennett cell compared to a lysate of a stem cell. This probably has something to do with all the other competition that's in there. But of course, if you do a frozen section, you will realize all this, and you can basically calibrate your data based on that. But we never did any. I'm not sure if there's anybody who did nuclei sequencing in the gut. If there's any good experience there, we always take the whole cell.
In terms of the comment you made about SOPs and the idea that, of course, you're dissociating a complex population comprising cells of distinct sizes. Some might be more epithelial, some more mesenchymal. So the rationale for developing protocols for dissociation, should one have multiple protocols, each that will favor any particular cell type that is underrepresented in another protocol? How do we address that challenge?
Yeah, very good point. So we are very much biased towards epithelial cells, because all these protocols, they basically get rid of everything else. And recently we wanted to see, the epigenetics I was talking about is very specific on the location of the epithelial cell. We wanted to test if the mesenchymal cells had the same thing, and it took us months before we actually were able to sequence the mesenchymal cells, because the protocols that we were using were the opposite. They were exactly the opposite you want to use for mesenchymal cells. So it's true. And then there's probably other cells that we don't even know of that just don't make it through any of the protocols. But yeah, every protocol is basically optimized for one particular cell type or one particular cell class, like epithelial cells.
Because one thing that you also have to be careful about is not recreate fax analysis version 2.0 in studying the single cell map of the gut. So if you keep sorting cells with surface markers, you probably go back to what has been done for the last 20 years. So having that spatial sort of cell-cell communication component, I think, is critical.
I just want to add to that. So we've tried a few different dissociation protocols for colon, and there's a dramatic difference in the fibroblast proportion. So some protocols will give you 3% fibroblasts, and others will give you 15 or 20% fibroblasts. And obviously the one with very few fibroblasts has a lot of epithelial cells. So I think it's really important to come to some kind of consensus, because that clearly, and also the length of the protocol has a profound impact on, the length of the dissociation has a profound impact on the data quality. So these are things that I think need to be harmonized.
Yeah, I always say this, Ben, I think most of the community does this, but now anytime you get a really interesting result based on single-cell sequencing data, you want to validate it with something that is not at all related to single-cell sequencing. So you want to do a stain of fish or immunofluorescent. You really want to do it with two different methods at least to make sure that it's not something that is, an artifact. There are false positives with single-cell sequencing for sure.
Hank, you have been quiet. Any thoughts on the gut atlas?
No, not at the moment. I come in late.
Others, Moshe? No.
So as experimentalist biologist in Aviv lab, Aviv Regev and Rami Xavier, I was playing a little bit with the gut, and it took me a while to get to a protocol in which I can say that I can isolate both epithelial and the non-epithelial compartment. But this is really a very big problem, and we need, of course, to test it and check whether the proportions that I get have something to do with reality, but I think we are close to there. At least for my examination, doing validation using fish and antibodies, I think there is we can manage to do something that is already something that we can say it's good. So
in the same prep. So in the same prep, you will have both epithelial and mesenchymal.
Yeah. So in the same prep, first I'm dealing with the epithelial cells because they are more gentle, and then I'm going back to the immune and stromal cell.
I think it's really important because it's really underappreciated how much these two cell types are talking to each other, and there's pretty very big phenotypes where you make a mutation in the mesenchymal cells, and then the epithelial cells do something very different. So there is a crosstalk, but it's very understudied.
Other thoughts?
So I have a question that's quite general, but of course it applies to this atlas as well, which is that, so there's a lot of single-cell RNA-seq papers published now, and a lot of them on similar tissues, and each paper has a list of cell types that are identified bioinformatically. But I have not seen a lot in the literature of people saying, I found these 40 cell types in this tissue, and someone else found these 38 cell types in the same tissue, and how do my 40 cell types correspond to their 38 cell types? And so I was just wondering how, as a community, we're going to define standards to have a consensus list of cell types that, you know, we believe is seen in everybody's data set or most people's data sets.
I think that's exactly the goal of the HCA, right? So that you need a lot of, you cannot do that by a single group. Everybody has to basically use the same standards, upload all their data to the same system as we saw this morning, and then you can click on these things and start to compare. I think that's exactly why we're here, right? To really coordinate this. But it needs a lot of infrastructure, right? You need then to have access to all the data without having to go through every FASTQ file at a time, right? It just has to be quick and all in the same format. So I think that is the ultimate goal of this exercise. I think here hopefully that will happen very soon.
I think the annotation that was discussed this morning is really crucial, and out of our own experience with some of the immune cells, when we show the data to immunologists or others, they say that can't be, that cell type is not present in that tissue. And that is an annotation problem in part, but in part it's also that very often, and I think you mentioned it before, or Ramnik, if you use profiling or FACS sorting on the basis of cell surface markers and current knowledge, then you confirm current knowledge. But if you go in blind, basically index sorted, then you can get to a large number of new findings, and now you need to validate them, or you need to find them back. And you don't find them back in what other people have been doing on the 40 cell types that Sham just mentioned. So how to solve those problems, how to solve identity, how to solve annotation, and what is the annotation now? If it is a mix between different, or it looks a bit like a T cell, and it looks a bit like an ILC, and now what is it and what to name it? I think that's a major problem.
And another thing which came out of this recent meeting, this single-cell genomics meeting, is that it's actually not that trivial to compare two data sets from different labs, right? So they have this, what they call CCA, it's like canonical correlation analysis, which allows you to really compare several data sets from different labs and even from different organisms. So you can, for example, take a mouse pancreas data set and compare it to a human pancreas data set and align them, and what they call anchor the cell types, and then you can start to basically deform, you know, the gene expression space in such a way that you can compare these things. And it even works for different organisms, but it works much better, of course, if you have the same organism, but then you can do this across labs. And then if you see a cell type missing in one lab and the other lab has it, you know, then you can ask yourself, perhaps they used a different dissociation protocol or whatever. But it's also, so it's not only the HCA that is going to do this, but also new methods have to be developed that allow you to systematically compare different data sets, and that's not trivial.
One issue with that, though, is that CCA works really well when you have the same cell types in both, but if you don't, it doesn't work that well.
Could be. Although I saw in some of those plots, actually, I saw it because one of our data sets was in there, you know, we were colored red, and then I saw that the red was very nicely mixed with a lot of the cell types, but we also had an island which was only red, you know, which means that we only saw that, so I have to figure out what that was. It could be a cell type that was, you know, contamination in our case, or we had a special, we were lucky to have a certain cell type. But I saw that there were actually clouds that were isolated. So that might be a cell type that they couldn't CCA on. But yeah, I don't know. it seems to me they're going the right direction, though, in kind of merging these data sets. And there's a lot of systematic stuff that changes from lab to lab, and it's amazing how these algorithms can just throw that out.
As an alternative to that, too, there are other ways that don't involve necessarily merging data sets, but transferring information from one data set to another. We and others have algorithms at this point that we're trying to get more visible with the Human Cell Atlas, where we can find latent space or reduced dimensional representations of features, and independent of how any other cell type or property between data sets, we can very quickly and rapidly project these features, these representations. So you can imagine having a gene expression profile that describes position along a p-axis as a continuous feature, and just very quickly being able to interrogate a new data set and transfer that information.
There's amazing stuff happening also in this artificial intelligence area, where you can, of course, use deep learning and then these latent spaces. I just heard about it, and I'm very much learning still. But it seems that there's a huge community in machine learning that can be kind of moved over to single cell sequencing.
So, and just extending this towards specifically the gut, if there are issues with the geometry and the sensitivity and the fact that we have to have different dissociation protocols, and it seems, I am not a gut person at heart, but it seems like it's a particularly sensitive and hard tissue to work in, it would be reasonable to generate these kind of latent space informations for properties, including cell types, and then use those to compare across disease versus normal, or different patients or different samples as well. Like, this would be a tissue that seems very rife to benefit from that.
Forgive my naivete on this, but maybe not quite an analysis question, but in terms of the spatial differences along the gut, is there any indication of how fine the spatial resolution of these different cell type domains would be? Are we talking about, say 40 microns square, or could we actually block the gut into, say, millimeter cube units?
I don't think we know the answer, but what my thought is that there's like little islands in the gut, that the line the gut, and what I'm thinking is that those are clones from early development. So what we can do in the gut is, you perhaps know, if you dissociate the crypts, you get this what we call hands, which is like a villus, and then there's like little crypts hanging on 5 crypts hanging still on this villus. So you know that these 5 crypts are very, very, very close, and then you can sequence those 5 crypts individually, and you will see that 4 of them look identical in terms of the methylome, and 1 is slightly different, or is quite different. So I think that there are patches. I'm not sure how big they are. I would say they're probably the order of, 10 crypts, something like that, 5 to 10 crypts. And my guess is that it's a relic from early development. Actually, you're much more an expert there. But we don't know. But an interesting thing is it doesn't really depend. All the cell types have that same. Within the crypt, of course, lots of different cell types, but they have the same epigenetics. So they're basically very difficult to see the difference between a stem cell and a differentiated cell, which was already published by other people in that same crypt. They basically have the same epigenetic properties. But then you go like 100 microns to the right, and then it's completely different, which I think is a relic of early development. And they just remember that. But I can't prove that.
Again, my question is more disease-related, but how about having matched normal samples as an internal calibration for the diseased tissue that we are looking for? What's the thought on that?
Yeah, so we've, so at least for ulcerative colitis, we've done that to try to compare inflamed versus non-inflamed, but also normal versus inflamed. I mean, that again, I think it's, the non-inflamed is, a relative definition, because there could be states of inflammation that's resolving over time, and one might not be able to know that until you get that information. I mean, the one sort of question that I've had for a long time is that, is it possible that we'll identify LGR5-like markers for the 50 or 60 cell types that are in the gut? Or are we going to rely on number of genes adding up before you define a cell, a tough cell, or a goblet cell, or a innate lymphoid cell? So that's one question. And then the second is, you know, how can sort of, I mean, the mucosal immune system has unique properties that, you know, we should be able to exploit to better understand how the mucosal immune system, I mean, in this case, it can be gut as well as lung, but how this works in the same individual sampled to look at the peripheral immune system. Because, you know, just studying only the mucosal immune system in that index gut patient is only going to give you part of the information. It would be nice to see in that same setting, you know, what's happening in circulation, what's happening in the lung and other organ systems within normals. Other thoughts?
I don't want to make it more difficult than it already is, but there's this very beautiful experiments from Hans Clevers where he shows that there's particular endocrine cells at the bottom of the crib, and they're expressing a hormone, you know, hormone A at enormous levels, so they're very, very positive. And then they move up, they switch off that hormone, and they start to switch on the other hormone, which is completely different, and switch off the first one. So actually, as these cells are, you know, living their lives, you know, from the birth to the end of their life at the tip of the villus, they actually might be able to express different hormones sequentially. So they change cell type. You would say, well, that's an M cell or whatever P cell. They are actually, during their life, there are 4 different cell types. So when, of course, when you do the single cell sequencing, you will just take a photograph of one particular phase, but the day before that cell was actually doing something completely different. So it's very interesting, but also complicates things, of course.
I just wanted to go back to the issue of harmonization across data from different places and ask whether, so I'm a statistician, so I don't actually know what all the challenges are associated with this, but is it possible, and are there plans to do the same samples? Obviously we can't do the same cells, but somehow, in some sense, very similar samples in, you know, swapping them among laboratories, because that would be very valuable data for trying to see how well harmonization methods work, etc.
Yeah, no, I think that's an important, I mean, so we did this for the microbiome, where we got 7 or 8 labs who were processing microbiome samples in their individual lab, and then got each one to share their samples. And different ways of, you know, data generation alone, no one matched the other lab. But the only good news was that when you looked at abundance of organisms, you know, they were recovered independent of the method of isolation or method of data generation. So it's possible that similarly you might find big picture patterns but may not get to cell specificity when the rules break down.
Well, we would definitely expect, from what we know about technology like this, there to be strong batch effects, but not only will it tell us that they exist, but that kind of data can be helpful in trying to adjust for those effects as well. So.
So there's also a large pilot experiment going on from Barcelona. Holger Heijn is doing this, and he's sending actually LGR5 cells from the same animal to multiple labs in Europe, and they use their favorite sequencing method, and then all these data sets are compared. So this will, and there's several labs that have the same sequencing technology, so you will see, you know, is there any batch effects within this technology, same technology? Does it depend on which lab it's doing? So I think we will also get some very good statistics out of that. But they come all from the same animal, so that they should be pretty reproducible.
So are they doing the control to send the same animal to different labs to isolate the LGR5 GFP positive cells?
So they dissociate it for you, which is easier, right? Otherwise, otherwise you will have much more variability.
So I think one more thing to follow on some of the topics before. In machine learning, we have these deep learning methods that are coming online, and the latent variable models that are coming online as well to be able to analyze these data. But I think we're severely lacking a bunch of different types of analyses that are essential to thinking about gut in particular. So spatially resolved, either deep learning or latent variable models are going to be absolutely critical. I think statistical testing is going to be a big deal, not just differential, but more gradient type testing is going to be really essential, even thinking about healthy versus diseased samples as well. So I guess there's a number of challenges that I still think that we need to start attacking methodologically.
Other thoughts on analysis methods? Maybe I can put Aviv on the spot. Since
I wasn't here, so I have no idea what people were going to say. Sorry.
You can highlight 3 challenges in analyzing single-cell data
from the gut. So the biggest one is that the gut is not as, it's the opposite of what Stan said about the brain. It appears, right, very repetitive, and it is extraordinarily long. So it might be that it's all really the repetitive unit, a few anatomical locations, and a gradient. That would be a really good solution if this is actually what it is, in which case I think you would get great answers easily computationally. It could be that there's stuff there that we are not appreciating right now, in which case it would not be so great, and we'll see what Barbara does. So that's I think, one layer of complexity. The other layer of computational complexity is that this is a very dynamic system. One would expect states to play a very major role compared to types, for example, whereas in the brain, again, it's a little bit the opposite of the brain in that sense. In the brain, you have this strongly typed cell subsets, even if some of them are organized on gradients and not in, they could be organized in different ways, but they're invariant with time. Whereas in the gut, you have a lot of stuff that varies with time, condition, and so on. And as disease kicks in, it gets even worse, but even without this, it's a dynamic tissue. So I think that would be a second challenge. So knowing where you are is the first one, and knowing that in parsing out this type versus state is probably going to be the second big challenge of the gut. And I think the third is the microbiome, that in this community, people are aware of. They're not disliking the microbiome, but they're also not paying a lot of attention to the microbiome, and it is an important component. And I'm going to add a fourth, which I think is true across the atlases, is that maybe initially when we thought about building those, then for most things we said we would look at relatively small cohorts. That's still doable. We know for full atlas building, that's okay, but we probably want to dabble into diversity, and we need to understand how you do that. And I would actually posit that in that the gut has an edge over the other organs. It's actually the opposite of the brain. There it's tough. In the gut, it's easier to get sampling from people, easier to do all sorts of things. But figuring out how to design for that is going to be very different, because the history of developing methods for relating genetic variations with traits has typically been for traits that are measured on average, such as bulk expression profiles or anything actually, average level of something in the blood. And here we're looking at distributions and all sorts of other types of statistics that we haven't thought about in the context of human genetic studies specifically. And the geneticist should correct me if I said something atrociously wrong, but I think it's roughly correct. It might be that the problems were very difficult for human genetics of quantitative traits will end up being easier in this sense, and the problems that were easy will turn out to be difficult in this context.
To follow up on what Aviv says about a highly dynamic system, it's also, we assume there's a stem cell with the progenitors and the fully differentiated cells, and in the gut, I think it's, everything can be upside down, Yeah, no, that's related to your dynamics, right? So they've shown that like an enterocyte, which should be fully differentiated, can go back and become a stem cell and do whatever a stem cell normally does. So I think we assume it's like the hierarchical kind of textbook thing, but it's probably much more dynamic and plastic, and we think it is, which makes it even more challenging.
So a lot of the discussion so far has been on expression data. And so what about other types? I mean, Alexander briefly mentioned about some of the methylation epigenetic profiles. What about protein, imaging, and all of the above? Metagenome. So let's go ahead and say something.
I think the gut is super interesting in terms of the different modalities of measurements, partly for the reasons of access, but also that the importance of interaction with metagenome, with metabolomics, interaction with diet, and so on. So Yeah, I think it's well worth considering, you know, how can cell atlas modalities be integrated with other I'll say the conventional cell atlasing, in other words, single cell RNA sequencing, the spatial methods, be integrated with not only genetics but other measurement modalities.
Yeah, so I mean, I can give you so I saw a patient a couple of weeks ago who was waiting something like 6 months to see me, and he decided when he came to my office, he fasted for 3 full days. He thought I was going to be able to identify everything that was going on in him by fasting for 3 days. I told him that I can't even do a single test on you because you fasted. But I think that's something to consider. I mean, how diet, metabolism, all of the above. Aviv?
I'll say several things. I guess Alexandra said something about epigenomics earlier. I think it is very important to obtain epigenetic data of the type that has much longer stability and timescales. It complements really well RNA levels in the sense that it actually reports on a different timescale, and it helps a lot with these questions like where things are coming from and where are they going, in ways that the RNA alone leaves you in. It can give you a lot of signal for inference, but this gives you somewhat orthogonal information. And it is great for human genetics because of the non-coding variants. The second item I wanted to highlight was the extracellular matrix, which has implications both for measuring the importance of measuring proteins. If you measure both RNA and proteins, then for secreted proteins, you have information about where they went to, and from the RNA, you know where they came from. So that's a unique combo that is very interesting. But also more generally for the extracellular matrix to have in situ assays that assess it well, that don't destroy it and so on, because a lot happens there, and it's a very important and structured component of the system. Sarah already highlighted the metabolites and so on, so I'm just not going to repeat it. I do think they're actually really important.
Other thoughts? Other thoughts? No, I'm going to have to pick on people who can stay quiet. Oh, sorry. Yeah. Question and answers.
So thinking about the ECM and also interactions with the microbiome brings me back to, I think, a technology point that Alex touched upon at the beginning, which is either computational or direct measurement of interactions between cells or cells and organisms. And I'm curious what people, so I think there are cool new methods on both sides, right? So computationally predicting pairs of cells or communities of cells. Similarly, now methods that directly capture those pairs, and whether, just to kind of look down the road, do you see how those two can validate each other, and or whether there's going to be specific challenges within the gut around the analysis and extracting that information or measuring that information?
In the same meeting I was mentioning before, there's a lot of paired cell sequencing was shown, which I think is a very nice way to do it. So you just basically dissociate the epithelial not fully, but you make sure there's still little pieces in there, and then you sequence them as a whole, and you can infer what are the members of that triplet or doublet. I think that is, it's a straightforward experiment. It's not very complicated. So I think we should do more of that. I think that is very helpful. The computational inference is, of course, you don't need, the data is already there. You don't have to do any new experiments, but it's always an inference. So although it looked very good, I think it works for large gradients where you have large-scale gradients. It probably works well. If you have a Paneth cell next to LGR5 cell, that's a huge change in the transcriptome. So I think you always have to validate these type of methods with paired cell or triplet cell experiments. And combining it with in situs, Murfish, all these things are, of course, you know what to look for, because we know these cells already. They're sequenced to very deep, deep level, so you can make very nice libraries, Murfish libraries, to really look at the statistics of neighbors and nearest neighbors. So I think that's the way to go, and at the same time develop new computational methods to really reconstruct it. I think there's a lot of exciting stuff happening.
I'll add to that 2 things. The first is the difference between the just molecular characterization of who's next to whom versus the inference of what they're doing to each other, where I think there's great opportunities to also try and predict what is actually happening, which is a more functional view and is getting a little outside of the scope of the atlas, but is definitely within the scope of what we wanted to deliver to people as a downstream. So that's useful to remember. And the second is that there is a difference between neighborhoods that are very stable. And so you would actually always see at the bottom of the crypt this cell next to that cell, this one's the very bottom, and that's at position plus 4, and so on, versus those that are more transient, for example, interactions with immune cells, because they literally are transient, they come and they go, but the style of the interaction repeats itself. And in order to see that, you need to look multiple times. Either you're looking at a big section, so you have a lot of places to see it, or you're looking across multiple specimens, so you get a lot of opportunities to see it. And those interactions matter too. And it's not clear yet, right, how many of them are stable or they're just a one-off, and finding the right, what is the statistical characterization of these types of interactions, I think is going to be a very interesting thing. That's different than the canonical structure. The stroma always sits here, the epithelial layer is there. Within the crypt, this is at the bottom, that's at the top of that's different.
Yeah, related to that, I mean, I'm very influenced by Hans Clevers, of course, because he's my neighbor, and he has these beautiful movies where he shows, you know, these cells being born, and then you have this conveyor belt of cells that go one by one to the top of the crypt. And then you say, okay, that's how it works. But of course, it doesn't work like that at all, because you can do lineage tracing and you can show that different cells actually have different velocities going up. So that completely violates a conveyor belt, because they're basically passing each other. And dependent cells, they escape it. So I think it would be nice if somebody can make a movie of a crypt and these cells show how these cells actually move, because I have no idea how this works. It is very interesting how this happens, because we know from other experiments that cells move at different velocities. So that cannot be a conveyor belt. Perhaps there's different conveyor belts next to each other with different velocities, and they mix a little bit. But anybody can make a movie, a real movie, not like an animation. That would be really helpful.
I mean, so this kind of dynamic process obviously also happens during development. I mean, have you discussed, because I missed the first part, have you discussed sort of human developmental stages? And that's relevant, you know, okay.
One topic before we get to development, so we said 30, 40, 50 cell types in the gut, but there are some cell types where you cannot get transcriptomics data, or at least with the current methods, it's a lot harder. Any thoughts about that?
What, you mean neutrophils?
Yeah.
It depends on the tissue. I mean, because it depends on the tissue, I would say. I mean, because in bone marrow,
Yeah, so for example, you can't, neutrophils, eosinophils in tissue is almost impossible to get transcriptomics.
I think it depends on the tissue as well.
So esophagus, colon, or small bowel, it's hard.
Yeah, I mean, in lung, they are actually efficiently captured. So it depends a bit on the tissue.
Lung is not, we decided before we started that the gut is the most important organ.
I think that does bring up something that we really can learn from the other organ systems. Like, because I'm sitting here and I'm listening to all the challenges, and they're almost the same challenges that you have for the skin, right? It's really, really big. It has a microbiome. It interacts with the environment very dynamically, the same way the gut does. So yes, the gut is the most important organ in the body. Coming from where I come from, that's definitely true. But we should still keep an open mind and keep an eye on what other people are doing in other areas of the body, because I think we can learn a lot of lessons and learn from each other.
Good. So
moving on, the in situ method should also reveal that.
Yeah. So maybe the one topic that we haven't touched upon is developmental biology in the gut. Maybe Sarah can start, and then I think you mentioned you're interested in some aspects of development.
Oh, you are just talking about development.
Yeah. Lineage tracing.
Okay, that's fine.
Yeah. I keep coming back to the idea of lineage tracing in the gut, which I think would be incredible. And where do we start to do that within the human cell atlas? And can we do that within organoids? And can we also consider bringing xenograft models into this as well to create more of a dynamic human cell atlas? So does anybody have any thoughts? I'm a fan of viral lineage tracing. But applying this to human organoids, I think, will be challenging as well, and need some modifications of our current system.
Yeah, we're very much interested in lineage tracing, and we're very much interested in the gut, so it will be a good combination. Of course, you cannot use CRISPR for this. If you want to, it will be difficult. So we are thinking about endogenous ways to do lineage tracing, and that's actually why we started with the epigenomics. I mean, this is, when I was mentioning that it might be a relic from the early development that is, in a sense, a lineage trace. So DNA methylation is one of my, candidates to use it. Of course, you can also use somatic mutations. And we're working on that as well. That is a big needle in a haystack problem, but if you do it right, it works. But it's challenging, of course. So I think those are the two things that we are thinking about for lineage tracing purposes: somatic mutations and DNA methylation, both at the single cell level. But that's a lot of method development at the moment.
Yeah.
When there's like clonality, right, coming from the stem cell, do you think that one or the other, the methylations versus methylation or another epigenetic mark compared to somatic mutations kind of has an edge over the other? Or you would only ever be able to say that's one crypt, that's the next crypt, but you wouldn't be able to say something within the crypt?
Yeah, so of course the crypts are a nice system because they're probably clonal, right? So that's why we start with it. But what you can, for example, do is take, so we now work with whole crypts and analyze those, but then we can also dissociate them and take half of it, do single cell work, and the other half do semi-bulk. So it's a very nice system to actually figure it out. So hopefully you can deal with whole crypts because then your signals will be much better. Single cells will work, but it will just be more noisy. But we can prove that it's feasible, right? So if we just analyze enough single cells, compare them to the crypt they came from, both methylation and DNA somatic mutations, then we can hopefully, what I'm hoping is we can just deal with crypts. That's the hope.
Yeah. I mean, we've put on bioRxiv a method called Cardellino, so shameless advertising for integrating deep exome sequencing with single cell RNA sequencing. I don't know if that's kind of the direction that you were thinking, but I think from a technology and methods point of view, tackling lineaging in human tissues in development, like there, in theory at least, it is a needle in a haystack, but in theory at least that kind of approach should be applicable.
Yeah, one thing we learned is this is perhaps a little bit technical, but that phasing your somatic mutations to existing SNPs is making your life 10 times easier. Yeah, so whenever you want to do this, you want to make sure you have two genomes that are quite different from each other, like a hybrid system, which in the humans, I guess, that's going to be a little bit more challenging, but it's really important for calling the somatic variants.
Well, I just felt that there's a lot of outstanding questions, and there's a lot of disease relevance in terms of, for instance, diseases that manifest themselves postnatally very, very early. Presumably, their origin, the origin of the penetrance, can be already during fetal development, right? So from that point of view, understanding development is going to have an impact on the clinical side too.
Other thoughts, comments?
We've talked a little about lineaging, and I think there's a lot of interest in performing lineage tracing. So could we perhaps maybe shift a little to morphogenesis? So how do you, over time, during development, generate the form that we recognize in the adult? Any ideas? And how does one tackle the overarching problem of sort of building sort of temporally sort of your three-dimensional context? Because the gut doesn't start with the villus script structure.
So I think one huge challenge here is, of course, that you have beautiful work in the mouse. We saw it as well in the last few days. But to do the equivalent experiment in the human, it's impossible, I guess, right? To get that gut tube at that stage of development, how would you ever get that, right? Is that possible?
I think were the embryos to be available, I think the later stages that we've done with Dana pair, one could obtain, and I think the protocols, and again, back to the protocol development and the dissociation. So we've essentially, with Dana's lab, performed a study on the early gut tubes of sequentially staged embryos. It took 4 to 6 months to optimize the protocols to isolate the tissue and then to dissociate the cells. And for every stage that we looked at, we had a slightly modified protocol. So effectively, I think were one to be able to get the embryos, and one in principle could, one could adapt the protocols that have been optimized in the mouse to microdissect out the tissue. The reason we did that in the mouse was that if you take a mid-gestational embryo, it comprises around 200,000 cells or more. The gut tube, or the rudimentary gut tube, is less than 3.5% of the total cell number. So that's about 8,000 cells. And because we wanted to sample about 50,000 cells, we had to optimize the isolation of the tissue. So I think that could, in principle, be done, and would rely on having optimized protocols for the sort of enzymatic isolation of the tissue and then the dissociation. So I think those later time points that we looked at, one could, in principle, get some sort of, you can have access to human at that stage.
And in that case, the right design for that would be basically to do a time course of the human.
Totally, yes. And I guess the other, again, idea that we're thinking, and we're developmental biologists, we work predominantly in the mouse, but I think what will apply to the mouse will provide the framework of how to move forward in the human, is once you can isolate the tissue, could you ultimately use it to seed in vitro explant cultures to look at, in our case, we're very interested in the outpocketings of different, not the gut, but the anterior, which does outpocket and form the organs. And I think that's going to be something that in principle you could, if you could isolate the tissue, you can do the in vitro culture. You can get at least a couple of hours and some temporal information, and ultimately start to build detail also on the spatial sort of branching and initial morphogenetic movements. Yes.
Okay, so I think the last thing on my cheat sheet here is to talk about gut and disease. So if you take organ systems and look at textbooks of medicine, gut is the organ where there is most number of diseases of unknown etiology. There's a few examples like celiac disease, and Helicobacter pylori causing gastritis and gastric ulcers, but 90% of GI diseases have an unknown etiology. So they come with a symptom complex that then localizes to a specific organ, and then it gets a name, like in medicine. When a couple of symptoms add up, you call it a syndrome. And when it's seen in other organ systems, you call it by somebody's name who described that constellation of features. That is human biology and disease classification. I'm simplifying it. But I think but I think there is a unique opportunity. So the other side of the coin is that the GI system is susceptible to infectious disease, metabolic disease, autoimmune disease, cancer, neurological disease. I mean, every type of etiology can affect the gut. So you have this unique opportunity of seeing, how these other processes might contribute to what's going on. So I think I warned everybody by saying, when you study the normal patient or the normal individual, you have to make sure, because you tend to pre-select for people who are interested in the GI system who volunteer as normals. So that's not a true normal. So I think that has to be taken into account. Disease, how does one go about this? Obviously, with every disease, the initiation of the disease is hard to capture unless you follow a cohort of people who are genetically at risk and then wait till they get disease. But are there other ways of doing this? So obviously in diet, one can do this. You can sort of see how obesity develops over time and how the gut might adapt to this.
No, I think Ramnik raised many interesting points. In particular, the gut's contribution to diseases in other tissues, such as neurologic disease, and should one be, doing atlases from every tissue from diseased individuals to try to understand, the multidimensional contributions of an overtly normal intestine to, for example, Parkinson's or neurological disease. With respect to diet, again, diet is a contributor to, many aspects of human disease, including the intestine, and trying to dissect out the role of, aberrant pancreatic disease that arises in obesity versus the microbiome versus cell intrinsic changes within the epithelial lining itself. The immune system, again, raises the question of whether we should be, doing these types of atlases from the same patient from multiple tissues.
Yeah, so getting multiple tissues from the same individual, I think, is going to be critical because you're not going to be able to know organ communication. Now, getting back to so again, another piece of evidence why the gut is important, because according to Science Translational Medicine, Parkinson's disease starts in the appendix. Any thoughts? So maybe I'll put the Helmsley Trust on the spot since they're here and they want to know Crohn's disease, what would they like?
You're putting us on the spot. That's great. Yeah. So, I mean, the interest, first it's that, as we announced earlier, we have an RFP that is announced in partnership with CZI. They're looking at the seed networks. We are trying to build the gut cell atlas. And we try to focus actually, and this summarizes in a way some of the conversations we've had earlier related to location of the disease or location of where we have to start. So we have to start somewhere. And for us, because the disease is really, it affects the entire gut. However, big percentage of Crohn's patients, they have the disease in the terminal ileum and ascending colon. And this is where actually we decided to start the gut cell atlas. And at least from our perspective or our approach. And by doing that, actually, we probably will be able to focus and be able to compare across different institutions, across different labs, and replicate the discoveries, which is something we've also touched upon. So now the disease, which you know very well, Ramnik, is very complex, and it has different phenotypes. So from one patient to another, we might get to yet another challenge of heterogeneity that it will be very difficult to explain. So maybe the genetics has a big component, so this integration of the data and trying to sort of have a multi-layered approach to study the disease and comparing to healthy tissue. That is something that I think it's important to focus on. And imaging has, I think, a big component because paired cells or working with sort of trying to study single cells versus paired cells versus a group of cells, having imaging to back up that and to sort of build the entire structure of the gut, even having endoscopy imaging, which could be extremely helpful in some instances, because now there are more and more technologies that can really go deep and even start looking into sort of at the microscopy level imaging of the gut and have this 3D structure. So, I mean, this is just open for discussion, but for us, it was extremely important to start from somewhere and hopefully expand on. I think Oni mentioned earlier the creeping fat. Creeping fat is a signature for Crohn's disease. We only see this type of development of the fat tissue surrounding the gut in Crohn's patients. So having sort of even going deeper in the tissue and studying even sort of cells that one might not think of studying, it's extremely important for us.
I wonder if one way to distill out, the role of the intestinal lining versus, interaction with the immune system or multi-organ communication would be to, to take a step back and go back to animal models, but chimeric animal models where one can, for example, propagate organoids and repopulate the intestinal lining of immune-compromised mice or humanized mice to start to decipher, what role the lining itself might be contributing to the overall disease phenotype one observes. I mean, I know the Gut Atlas, the goal is to, characterize diversity among, diverse organs and tissues, but to get to more mechanism, maybe taking a step back to chimeric animal models might be helpful.
Could you expand more on that? Because initially when Hans and others started sort of generating these organoids from the gut, the idea was actually to transplant these organoids back, and mouse models were used for that. Unfortunately, we're not there for human. So how can you explain?
I mean, I think, yeah, sure. So I think, one possibility is, one, from biopsies derives, expands organoids and, reepithelializes the intestine of, recipient animals, and then, for example, tests out the effects of diverse types of microbiome on those engrafted organoids in vivo to, for example, understand what role different microbial communities are contributing to the overall epithelial phenotype. So if the hypothesis is that, if this is a multidimensional process, how does one break it down into simple components? So one way would be to epithelialize, recipient mouse colons and then introduce variables within the microbiome to assess whether this brings out for example, a Crohn's phenotype or another disease phenotype one is interested in studying.
I can speak to this forever, but just to highlight that these are things that are being done now in the field. So taking epithelial cells, generating organoids, there are lots of systems that have been developed, 3D, 4D, 5D even, combining all the different types of cells, sort of in the luminal side versus basolateral side, where you put immune cells and see how the microbes interact through that barrier that you're building. So gut-on-chip models or others that Hans, many other groups, actually they've been working on. So these exist and they are happening in mouse models, but also in human. So they will help us a lot and guide us through the process. However, going back to mouse, when we're building the human gut cell atlas, I think it's just taking a step backwards rather than moving forward, identifying, regardless of the heterogeneity we have in humans, how can we really study the human gut? So I mean, just our perspective.
Can I say something real quick about mouse models or organoids? I think fat cells are really hard to establish in organoids. You can get epithelial cells, same for mouse models. I mean, I don't think there are any models for creeping fat in either systems. So if there are any, or if anyone can build fat cells in organoids, that would be pretty awesome.
Just two random thoughts. I mean, one is I would sort of turn it on its head and basically say that, for understanding these systems and their fidelity, the gut cell atlas will really be, extremely interesting and useful from that point of view, I hope. And for informing these systems, and so maybe we can look at it from that perspective. And then the other thing I was going to say was, so with the interaction with other systems like adipose and fat and so on, it's hard. The deceased transplant donor program that I mentioned in the talk this morning could be useful from that point of view, because you can access, you can kind of chop through deeply, if you see what I mean, the tissues. You can look at multiple different regions, even different organs, in, sort of in parallel. That's another thought. Yeah.
So they're obviously trying to get to a disease as complex as Crohn's disease from normal requires many steps. But there are also some key sort of histological features that have been described in Crohn's disease since the 1920s. So things like creeping fat and the idea that disease, when German pathologists looked under the microscope, always started in the vicinity of a Paneth cell. So I think there are these long-standing concepts that one could sort of revisit, obviously with much better resolution.
Yeah, just to add on that, actually, another complexity of Crohn's is that it's patchy and it's skipped lesions, meaning that comes back to earlier conversation that these patches or these crypts that are 10 or 15 crypts, I don't know how many, that are in pools, maybe they're behaving differently, and maybe this is why the disease skips in some areas of the gut. So there's benefit of understanding all this, and I think the Gut Cell Atlas will allow us to do it.
So I'm going to ask a power question. Is there any sense, I think a critical question for really assembling an atlas, both for the healthy side and definitely for the disease side, but it applies to both, is how do you do the power analysis for sampling the system? And that goes to how many individuals, and then within an individual, how do you sample? I mean, the thing is big. You're not going to actually look at all of it. So knowing how to sample it at different length scales is going to be very important. Maybe one of the statisticians wants to comment on that. Maybe.
I had a question along those lines, then you can comment on the statistics. But what I was thinking about was you have any disease, like you probably have several stages that you're going to want to look at, not just several places within a patient. And also you're probably going to want to think about what treatments they've had. And so all those things will come into play. So that may, in the end, limit how many diseases you go after or how deeply you look at them, because you may miss something if you don't see the whole course of the disease.
Well, I mean, in this particular RFA is only one disease. I can't go elsewhere.
The HCA is bigger than that.
No, I'm talking about
I think for us, it's critically important anytime we're talking about Crohn's disease because it's so heterogeneous that you have to have really well annotated metadata about the patient. Otherwise, you're going to have a really difficult time trying to compare data across data sets. So, back to the annotation.
Just to tag on that, so in addition to having the metadata, it is very important to always keep in mind whenever, I mean, at least for the RFA, to have sort of as focused as you can, the cohorts to be, or the patient population to be sort of an early stage or on one drug rather than 15 different drugs, because these patients undergo so many different therapeutics because the disease progresses sometimes very fast, especially if we think about pediatric. And it's extremely important to really have that information that is linked to the patient as well as the sort of the treatment, because I mean, I totally agree. It's going to change the entire sort of makeup of the cells, and that's important to have.
Other thoughts?
This is a question that came in from the channel.
Yep.
But it's not just as we're returning to disease, so it's. Can you hear me? Okay. I'll just read it. Beyond genes, what other molecular moieties would be of value to integrate into the cell types indicated by the single cell sequencing data? Is there interest in other RNA species?
Yeah, the more you can measure, the better. You know, that's I'm a physicist, so I love to measure as many dimensions as possible. So mRNA, of course, is the one which is really established right now. But there's a lot of other RNA which we completely ignore. So it would be very informative to look more broadly at RNA, microRNAs, obviously. That they will do something important. It's at the moment not very trivial to get those microRNAs out single cells, but we and others are working on that as well. But I think a lot of like boring RNA molecules, or what we consider boring RNA molecules, might be very important, like snow RNAs or small nuclear RNAs that regulate the splicing or the modifications. I mean, everybody says that they're all the same in every cell, but I don't believe that. I think there's a lot of interest in the molecules are super important, but it might also be very cell type specific. At the moment, it's limited by the methods. We don't have methods that work really out of the box that will give you all of that richness, but that's what we're working on, others are working on. And beyond, even ribosomal RNA, you know, it might be very interesting. So single cell ribosomal RNA might not be all the same. Nobody knows. And of course, beyond RNA, yeah, ultimately you want phosphoproteomics in single cells. That's what you really want. Again, it's all limited by technology. Like mostly in biology, the big breakthroughs come when these technologies start working. And mass spec, you know, I'm waiting for the first papers to come out, and then we'll implement it. But it's I think still far away.
Thoughts? I mean, since colon cancer is a complication of Crohn's disease, you're allowed to drift into that space.
Thank you. Actually, I was going to say something different, not colon cancer, but if that's okay. So before we discuss disease variation and so on, it was alluded to earlier today that we have to characterize the range of normal human variation. And this is really challenging because anyone who's analyzed multiple data sets will know that the cells cluster by batch rather than by cell type very often. And that's a bit scary because there's a hierarchy, right? So the difference between two major cell types should in general be different larger than the difference between two individuals in one cell type. But if the batch effect is bigger than any of that, then how are we going to characterize inter-individual variation? And just hoping someone has clever ideas for how this could be achieved. Because we really need to do that before we characterize disease versus normal, or to help us characterize disease versus normal.
I mean, I used to be very worried about that, like for human cell atlas or for single cell technologies in total, but I think there are so many nice kind of alignment, regression, integration methods nowadays of different categories. I mean, graph-based machine learning, deep autoencoder, transfer learning, whatever, that I feel much more kind of happy and optimistic that we can do massive alignments, massive integrations. When I say we, I mean the computational community, we and others. I don't know, Aviv, do you want to say something about that?
I would just say the same thing, and I generalize it. It seems that there's a good framework for both how you would combine things across conditions within a patient, across individuals, and also across data modalities. It's actually the same type of conceptual and mathematical framework that lets you do that. And it relies on the fact that there are some facets or aspects that are actually shared. And it finds those things without you necessarily having to actually tell it that it's there. I think what is interesting and not fully yet done, and there's a great opportunity around, is how you harmonize that or bring into this framework the prior knowledge. So, and that's important, and we see that, a place where you see that very aggressively pursued is in the brain, because there's a lot of prior knowledge and anatomical characterization of the brain that people feel very attached to for good reason. A lot of, decades and decades of research are associated with the fact that you call it that it's the MPOA, and you know which behaviors go with each nucleus there, and so you need to know that you're in that thing. So even if you could, in principle, rediscover it, you want to rediscover it and know that you rediscovered that thing that everyone called by that name and associated all of those things with it. And that requires bringing in this anatomical, subanatomical and histological knowledge that already exists. And I think that would be an interesting computational challenge, but absolutely doable. Yeah.
Requires the data, though. You need a volume of data for these problems.
So just from someone who's not computational, just to answer you sort of an easier way from my way, is that you know, if you want to understand normal, you have to have a disease to really say that this is the normal state. So just I mean, it's very difficult to say that a gut is normal, a cell is normal, if you don't have it in disease state, whichever disease you're taking. So I think last year at one of the HCA meetings, I think it was an example on the skin, where they started looking into disease state, and this is where you realize that maybe you have batch effects and then you can correct for those. Otherwise, it's going to be very difficult to really sort of be just inclusive or exclusive of some groups of individuals. We are tackling the disease from Hemsley's perspective at the personal level, but to be able to do that, you have to be inclusive, you have to include everyone. Otherwise, it's going to be very difficult to identify what's normal, what's disease. So just my simple way.
Going back to Aviv's point, I think the idea of prior knowledge is really interesting in how to incorporate that metadata, everything we know about each patient. So, you know, one school of thought says that we actually incorporate that explicitly into our model. We sort of bake it in and then, you know, hope that will add power to the results that we have. But another school of thought says that we actually hold it out and use it to validate the signals that we find in a sort of less biased way, if you will. And I think that it's probably worth thinking about when it's useful to bake it in and when it's used to use it for validation. This comes up in a lot of contexts, obviously. Any underpowered study is going to have this. Then going back to Sarah's awesome optimism about how the methods will be built, I think there's also a skeptic's point of view. I shouldn't say I'm a skeptic, but I'm a little bit of a skeptic because you know, I've worked with these data a lot, and the problems are really hard. And I guess, you know, even working with very quantitatively savvy people, there are some things that we still can't do right. So for the people who are designing experiments, I highly recommend having a discussion at the point where you're gathering the samples and designing the experiment, just to sort of gut check quantitative people about what is not understandable from the data that you collect. What problems can't be solved given the design of the experiment and the available methods?
Yeah, I was going to make a related point, which is that batch effects are really important to worry about, and but what really matters is, and what's a disaster, is if the batch effects are completely confounded with the thing you're interested in. So if you're interested in the difference between two cell types, it's a disaster to do one cell type in one experiment and a different cell type in a completely different experiment, and not to replicate that. But if you have the two different cell types are done in lots of different individuals across the world in different places, then hopefully some genes are going to be different between those two cell types consistently across all those replicates. And those comparisons within experiment are kind of, you know, valid, if you like. I mean, there may actually be batch effects within an experiment, but let's ignore that for now. You can do those comparisons across all these different replicates and look for what's in common between them, and that, you know, the batch effect effectively can reduce power, but given enough data, we'll find those differences. So yeah, just when you're designing experiments, make sure that the batches are not completely confounded with what you're interested in.
Yeah, I just want to add some optimism also here, because we recently sequenced quite a bit of breast tumors, and at the single cell level, and many of these are very patient specific. But what was very nice to see, and I didn't expect this, is that there's populations in these tumors that are very similar between patients. Like there's macrophages, for example, that you can use as a internal reference, and they basically exactly fall on top of each other. All the patients, these macrophages, are exactly the same, right? And so it's in the same data set, in the same prep, everything is the same. So you have a couple of cell types, which are mostly the healthy cell types, that are very reproducible between the patients, and that gives me hope that the batch effects are not overruling everything. And of course, there's lots of variability, but then you have a couple of these cell types which are really pinning down the high dimensional space, and you can be sure that it's not completely dominated by batch effects.
I will add to that this has now been seen across every tumor that anyone has studied in single cell analysis. That the normal, meaning the non-malignant cells, they can be dramatically impacted actually by the disease, but they're impacted in a canonical, stereotypical way that you see across patients. It's true that patients can then stratify. Some can have MACs or not have macrophages, or they could have in this state or in that state, but after a few patients, you're already in this very stable place. It's also true that people have looked by now at colons and at small intestines, and they again see good reproducibility. And so it doesn't seem to be as I think Sarah and Alexandra said exactly what everyone else thought in the beginning, right? That it would actually look much more like a hot mess than it ended up looking. Moreover, the techniques actually got a lot better at allowing you to reduce the batch, to bring it really, the patient is still a batch. You're not gonna control that. But everything downstream from that increasingly can be controlled in ways that in the initial days of these types of approaches you couldn't. So for example, there are multiple ways of pooling and demultiplexing later, meaning all of the experimental process, or from a very early step in the experimental process, it's actually done together, and then you can split them and mix them and do all sorts of lovely things with good statistical designs. It was not possible early on, and in tumor samples, it's rarely possible, because they tend to be analyzed kind of as they come along, and they still look pretty good. So not too horrible.
Okay, yep.
I wanted to add to Matthew's point about confounding of what you want to measure with the batch effects. I think for inter-individual variation, that is a serious problem. As Aviv was saying, if you can pool all of them, then you're substantially reducing the technical batch effect. If you can get your tissues from 5 people on the same day, more or less simultaneously dissociate and pool the samples. But that will often be impractical in many scenarios. And then you're in the worst case scenario where batch and inter-individual variation are completely confounded. And then what do you do? Maybe you sample the same person 3 times on 3 different months or something like that. Even that seems hard. So I feel it's still a hard problem.
We have some questions from the channel, but I wanted to make sure there wasn't any more comments or discussion about batch effects, because these are different questions. So,
yeah, that's fine.
Okay. Yeah. So the first is, I'm wondering how classification of diseases in terms of casualties and evolutionary explanations that Rusyan Medzhekov proposed factors into studying cells. If anyone could comment on that. Should I read that again? All right. I'm wondering how the classification of diseases in terms of casualties and evolutionary explanations that Rusyan Medzhekov proposed factors into studying cells. That's the first question.
Second question.
Okay. Second question is, can anyone comment on how to sequence neurons at a single cell level from the 2 plexuses in the gut, especially in heterogeneous presentation of disorders such as Crohn's?
I mean, I can answer that, but I think I'll give it to Aviv.
I'm sorry, I was trying to read the question because I couldn't understand the second part.
The second question.
Sampling the enteric nervous system. That's what nuclei are for. So it is difficult to isolate neurons as cells. This is established from studies in the brain from several years ago. And as Ramnik knows, we actually participated heavily in that. And several years ago, single nuclei RNA-seq methods were established by which you only isolate the nuclei and not the actual cell. And they turn out to generate very robust data. They have several additional benefits. For example, they can be applied to, actually they are applied to frozen tissue. And because they're applied to frozen tissue, you can actually archive your tissue and pull it and do all sorts of fancy stuff with it along the way. I would say that at least in our hands, the original protocol that we ourselves published for the brain works extremely well for the brain, but didn't work for the enteric nervous system. But after a fair amount of optimization, I actually talked about it earlier this week, after a fair amount of optimization, it worked very well for the enteric nervous system. What does not exist, at least today, for nuclei from the enteric nervous system is enrichment
meaning the ability to select only for them out of all the nuclei. And because neurons are about 0.05%, even in the part of the gut, in the muscular layer where they're relatively enriched, they're about 0.05%. It means a lot of other cells. There are other benefits there. The same protocol also recovers for you the adipocytes, the muscle cells, all sorts of cells that are difficult to isolate. I think they're a critical piece biologically, but Ramni can comment on that. Ha.
There's actually more.
Okay. Yeah. First question, I'm still not sure. I mean, so if I remember what Ruslan said was that, so I think Crohn's disease, though described as one disease, I think it's probably 10 to 12 different diseases. So there are many components or many different steps that contribute to disease 1, disease 2, and disease 3. And if you apply some of the concept Ruslan has been talking about is how those early controllers and regulators of that process might be different. And by studying those early events, can you predict the final clinical presentation of that disease? And so I think single-cell RNA-seq is going to help in that space. But I think there has to be some agreement about how many cell types are normal and abnormal in patients with Crohn's disease.
I'm not sure if I understood the question, but thanks for answering that, Ramnik. Maybe the question was more related to progenitor cells rather than evolution of the cells. So if it's progenitors, then the developmental work that is being done in the gut at early stages, as mentioned earlier, those will eventually define where these gut cells or the immune cells in the gut, where they're coming from, and same thing for epithelial cells. So where they generate and how they end up being in those different patches or groups of cells in that architecture, in that spatial.
I mean, there's also a possibility of additional sort of nature's experiments. So for example, it's now, I mean, there are a number of patients who don't have a history of Crohn's disease, who have underlying psoriasis, who have got treated for their psoriasis and now got Crohn's. So this is a nice example of how one might be able to track. I think I have probably about a dozen such individuals in my practice where these patients have been having psoriasis for 10 years. They went on the IL-17 drug and now have newly diagnosed Crohn's. So there you could hopefully be able to track the genesis of the disease from an early stage, and you have a perturbation in the system that one can track. Other questions? Sorry.
Can you comment on the potential solutions to optimally store and query obtained big volumes of single-cell RNA-seq data?
How do you store and optimally store big volumes of single-cell data?
We're planning it in the data coordination platform. What the final solution will be is an open question because we haven't got a large volume of data yet. So cloud, but yeah, it's in the cloud. So it's cloud. It's all going to be in the cloud in multiple cloud environments, both at least Google and Amazon, full stop, at the moment, both available to use in the cloud and to download locally if you wish to. Obviously, a move to bring an analysis to the data is a paradigm that everybody in the HCA supports.
If we see an extreme rare population based on gene expression patterns, which was never seen in bulk, what other level of evidence would be needed?
Yeah, so I think I briefly alluded to it. I mean, can you identify other LGR5 positive, LGR5-like markers for every single cell in the gut? Chances are probably no. And it's likely that there would be sort of broad phenotypes, I mean, which we've already learned from FACS sorting. There's a CD4 cell and there's a CD8 cell, but then if you want to create flavors of CD8 cells, it's going to have features of one and features of the other, and then you sort of defined a term called innate lymphoid cells a few years ago, but it's likely that there's going to be 15 different innate lymphoid cells and many, many types of CD4 and CD8 cells. So I think the single gene to cell type definition is going to become more and more blurred. The other point that I made earlier today was so you can find genes that are describe cells and cell states, but is there genetic evidence that that gene is important for that cell?
I was just going to go back to the first question. So correct me if I'm wrong, but LGR5 was first identified in the mouse and then by extension in the human. So what would happen if we identified LGR5 as a marker and didn't have the previous mouse data? How, I guess that speaks to the question that was alluded to. So if you didn't have the a priori data in the mouse, you identified LGR5 de novo in the human, how would you validate that?
Yeah, I think one of the huge pluses with Hans was that he identified LGR5 and then went to organoids. So he was able to track the importance of LGR5. But now if you saw an LGR5-like transcript in Paneth cells and you did not have organoids, it would be hard to know whether that gene is required for Paneth cells or not unless you make a knockout. But it's still not going to tell you the developmental trajectories of that particular cell type. Yeah. Are we done with questions? Because I think...
I have a question following on cats. What do people want in terms of validation, right? So there's a kind of level 0 validation is you said it's in this cell and it's actually in that cell and that cell actually exists. It's not a doublet, it's not an artifact of the experiment, it's actually in that yes, I get it. Is that enough or is there something else that we should be, obviously there would be many studies by many people who would use it, right? The analogy I like to use mostly to myself is that when people sequenced the human genome and they annotated the genes, they didn't actually, along the way, gave you their functions. In fact, we don't know yet, right? And it's been many years, and it served its purpose very well. So is that what, say, you or Garabet or Ramnik or Alexander would be looking for? Or is there something else that's kind of what's the minimal bar?
I think that's up for discussion, and we should get people's feedback. What is the minimal bar? That we haven't really talked a lot about imaging tools, spatial sort of distribution of some of the data that's coming out. Would an in situ or fluorescent in situ, is that useful information for us? I just wanted to play devil's advocate because I think this is a good forum. So what, any opinions?
I think the validation is going to vary a lot. So if you're talking to I often get these emails from Rich Loxley at night about, they found this gene in this particular cell type, whether I really believed it. So the challenge on an immunologist side, the evidence is you have to knock the gene out and that particular cell type has to go away. Whereas in the disease context, you could have grades of Paneth cells. So you could have a Paneth cell that's still a Paneth cell but not secreting antimicrobial peptides that could be disease promoting. And so that cell would undergo sort of the developmental steps that are expected of a Paneth cell, but its terminal differentiation step is where the roadblock might be. So, if you want to get to that level of resolution, then you need a new scientific discipline, I guess, to sort of track these things in vivo, and maybe you can make fluorescent probes that track your way all the way into cell types and light up these peptides and goblet cell markers and so forth. But I think the whole concept of the human cell atlas, Aviv and Sarah can correct me, is to define normal, move textbooks of medicine, immunology, whatever, a lot further forward, and then begin to understand human disease. I think we can stop there.
Perhaps what would have happened, so suppose Hans Clevers wouldn't be here, right? And we just would do this and we would sequence the human small intestine and we would see all these blobs in tissue space and say, hey, that's clearly a goblet cell because it's making mucin, and this is a Paneth cell because it's making lysozyme. And here's a blob of cells, and they we have no idea what it is, right? So in this LGR5 is like lighting up only in those cells. So then somebody would think, well, let's do the same thing in a mouse. And they do the same thing in a mouse, and they would exceed the same population, also expressing LGR5 and the goblet cells, they know, but there's this mystery population. And then, you know, some smart biologists would label it in the mouse, I think, and replace Hans Clevers, I think, right? Would just go upside down in that case, right? And so it would be very useful, I think, even though you don't know in that first experiment that it's the stem cell, but it's going to be a population that you cannot annotate, and therefore is by definition interesting.
I don't know whether I'm off base here. I'm going to throw the whole conversation in another direction. This one thing just keeps banging around in my head with different things we're saying. But what about, have we thought about connecting HCA to sort of the population genetics studies and whether, you know, unusual expression in cells, you could ultimately connect to that, and that particularly when we think about disease. Right, whether there's a use there.
Yes, that's already happening across many diseases. And I think we'll recommend to HCA that they should also start studying the microbiome. Okay.
Just as an advocate for the mouse, what about connecting HCA to the IMPC?
That's beyond my pay grade. Yes.
Which is the large-scale international effort to knock out every...
MC, or
yes.
mouse equivalent. And this is kind of the, you know, I mentioned developmental disorders and rare diseases. That's the mouse equivalent, kind of.
On the IMPC, Helen Parkinson, who's the EBI PI for the DCP, of course, is also a very important person in the Data Coordination Centre for the IMPC. So there are natural connections because we basically just are down the corridor from each other, which we should leverage better than we are at the moment.
Yeah, I mean, the goal is to make mice with all the coding mutations that are associated with human disease, and then we'll track every cell type. Okay, thank you everyone for 2 hours of discussion, and we'll keep moving forward.